Dear friends, good morning and welcome to another episode of my lecture series on MIT Homeopathy. To begin with, I have a humble opening request for you. If you find my videos anyway useful for you, please do not forget to subscribe my channel and kindly share the link to your homeopathy friends and groups. Today, let us discuss one of the most controversial and confusing topics in homeopathy, the issue of single drugs, which is considered to be one of the cardinal principles of homeopathy. In pharmacology, a drug is a chemical substance which produces a biological effect when administered to a living organism. A pharmaceutical drug, also called a medication or a medicine, is a chemical substance used to treat, cure, prevent, or diagnose a disease or to promote well-being of an individual. If a drug substance contains different types of chemical molecules that can act on different biological targets when introduced into an organism, as per modern scientific understanding, it is not a single drug. It is a combination or compound drug. This is only a very simple basic knowledge in pharmacology. Some homeopaths tends to forget, ignore, or deny this simple truth of scientific knowledge as they were taught otherwise by masters who lived and made theories 200 years ago during a period when scientific understanding of molecules, drugs, diseases, and cure were not available. We have to examine this single drug versus multiple drugs issue that confuses homeopaths by applying a rational and scientific mindset. Whatever our great masters have said earlier, within the knowledge limitations imposed by the particular space-time context, we will have to make a rational assessment of certain factors while trying to answer this very important question on the basis of updated scientific knowledge. According to so-called classical homeopaths, any form of drug substance used as a sample for proving is a single drug, irrespective of its molecular level constitution, even though it may be a complex mixture of several separate substances. According to them, the criteria for singularity of a substance is not its molecular level constitution or its biological mechanism of actions, but how its homeopathy proving is conducted. They think that even if they consume as a dose any number of different substances mixed together, it will act in the body as a single substance, as a single unit. This approach is obviously unscientific and irrational, beyond any doubt. Anybody with minimum understanding of modern science know that drug substances interfere in the biochemical processes of organism by their chemical properties, and that these chemical properties are determined by the individual constituent molecules contained in them. It is simple common sense that only because we consume different types of chemical molecules mixed together as a single dose for proving, or whatever you call it. It cannot act as single drug in the biomolecular processes inside an organism. For example, an alcoholic tincture extracted from the Naxomica plant is evidently a mixture of many types of enzymes, alkaloids, glycosides, phytochemicals, and diverse types of organic and inorganic molecules. It is obvious that tincture of Naxomica may differ in molecular constitution from sample to sample, depending upon whether they are prepared from the whole plant, flowers, tender leaves, bark, fruit, or any other part of the plant. No doubt, all these samples will be contained some molecules common to all parts of the plant, or what is called the species specific, even though their concentrations may vary in each sample. Over and above the natural organic contents, various elements and chemical molecules absorbed from the environment will also be part of the tincture. It may also contain various accidental contaminants and environmental pollutants also. In spite of all these possibilities, homeopaths argue Naxomica tincture is a single drug. Nobody can convince them otherwise, since our great master has taught them it is a single simple substance. Homeopaths should understand, when we introduce a sample of Naxomica tincture into a living organism for proving, its individual constituent molecules are absorbed, dispersed, and conveyed through blood and other internal transport systems into cells in different parts of the body. These individual chemical molecules interact with various enzymes, receptors, metabolites, and other biological molecules in the organism. These interactions between drug molecules and biological molecules are directed and determined by, controlled by their specific properties such as conformations and charges of active groups of individual drug molecules and their specific affinity towards biological target molecules. It is very important to note that Naxomica interact with different biological molecules not as a singular collective entity, but as individual constituent molecules and ions. These individual drug molecules and ions capable of binding to some or other biological molecules, effecting conformational changes in them and thereby inhibiting their essential biochemical processes, which can take place only with their presence and mediation. Such molecular inhibitions in various biochemical pathways result in a condition of pathology expressed through different traits of subjective and objective symptoms due to their involvement of various signaling systems, neuromediators, and neurotransmitter systems. The symptoms we get from the proving of Naxomica are in reality the results of diverse kinds of deviations in different biochemical processes and pathways created by the diverse types of constituent molecules in their individual capacity and specific conformational affinity. Each type of molecular constituent in Naxomica tincture binds only to a specific group of biological molecules and creates their own individual molecular inhibitions expressed through specific groups of symptoms. Suppose we could completely remove all molecules of a particular alkaloid from a sample of Naxomica before it is used for proving. Naturally, during the process of proving, we shall be missing the groups of symptoms that should have been created in the organism by the molecules of that particular alkaloid we removed. This type of proving may be called differential proving. By conducting such differential proving, by kicking out selected drug molecules, we can learn about the particular state of pathology that may be attributed to individual constituent molecules contained in each medicinal substance. Moreover, this type of scientific differential provings may be utilized to study the biological effects of various constituent molecules of drug materials that we erroneously consider as single drugs. Such differential provings may disprove our misconceptions regarding single drug at large. This is a subject that warrants serious attention from the part of homeopathic researchers and our premier research organizations. When drugs are potentized, the constituent molecules and ions are individually subjected to a process of molecular imprinting in water, alcohol, matrix. All the potentized drugs that we consider as single medicines are in reality a mixture of or combination of molecular imprints of diverse types of independent molecular contents and ions which can never interact with each other in potentized form. When introduced into the organism for therapeutic purpose, these molecular imprints contained in potentized drugs interact with biological molecules in their individual capacities on the basis of their conformational affinities. Since molecules and ions are subjected to molecular imprinting in their individual capacities and they cannot interact with each other in that form, there is no chance of happening any harm, any adverse effect, even if we mix two or more samples of different drugs in their post-Avogadro diluted forms. There is least possibility of any constituent molecule of drug substance remaining in their potentized form above 12C or post-Avogadro dilutions. Only molecular imprints of drug molecules will remain. Hence, when higher or post-Avogadro dilutions of two different drugs are mixed together, there will be no chemical interaction taking place between the molecular imprints. In such a mixture, the molecular imprints of constituent molecules of all samples will remain independent without influencing each other or interacting each other and without losing their own individual qualities whatsoever. What happens when such a mixture of two or more post-Avogadro potentized drugs is introduced into the organism of a patient? Naturally, the molecular imprints of each constituent drug interact with biological molecules and pathogenic molecules individually based on their specific conformational affinity. Biological molecules are thereby relieved from inhibitions caused by pathogenic molecules. This process ultimately removes the state of pathology and relieves the subjective and objective symptoms of disease. A homeopathic cure is said to be happened. Due to the specific conformational affinities, each type of molecular imprints can locate, identify, and bind to exact molecular targets, whereas in the absence of exact molecular targets having molecular affinity, these imprints will stay neutral since they are composed of mere water and alcohol molecules. The saying that if a homeopathic drug is not similimum to a patient, it will not act in potentized form is well explained here. The question of acceptability of administering two or more homeopathic medicines in potentized post-Avogadro form by mixing, alternating, or simultaneously applying should be discussed in the light of above observations. It is a very important fact that drugs in potentized form, which have no similarity with any group of symptoms shown by the patient, will not be able to create any sort of reaction in the living organism. This, I hope, I have succeeded in resolving the confusions and controversies related with the concept of single drug. Thanks for listening. We'll come back with another video next day. Repeating my earlier humble request once again. Do not miss to press the like button and also to share the link with your homeopath friends and your groups. Goodbye. Good luck.